Okay. Good morning, everyone, and thank you for the invitation to present here today. So I will give a brief overview of the current work that we're doing in the lab using combined approaches, vivo and in vitro, study the specific question around the topic of gastrulation. And I will explain what gastrulation is for those that are not familiar with. This is a moment in development that's quite important. And then we'll describe the in vitro system that we developed for gastrulation. They're called gastruloids. And this allows for modeling things that are happening in embryo, but of course in a much more high throughput. And hopefully I will show you a detailed way that you can really hardly do in embryos. And then discuss some of the questions that you can start addressing using this system. So this is very much in its infancy, it's early days. We've been working on this for a couple of years. There is a lot of progress and really exciting biology coming out of these models that have helped into some long-standing questions in developmental biology, really. And this is why I think it's a very, very promising area to invest time into. And then how we can use some of these answers, some of these approaches in medicine. So the translational side, just a couple of slides on that, not a lot of that, but just to give you how we're using this. So in terms of what the type of question we are dealing with, well, it's about understanding how cells emit different lineages in development. And so gastrulation is that period of time when the three major germ layers are established in the embryo, within the ectoderm, the mesoderm, and the endoderm. And there's a great variety of cell types in embryo. So we are keen to study this in the pig embryo. And the reason for using the pig is multiple, but to a certain extent, it has a potential application for generation of human organs. So founders like to see that there is a potential medical application to your model. And they also share big similarities with humans. And this is not a surprise probably to the audience here, but many people, when I talk this talk, communities such as very human or maybe rodent-centered communities, comes to a surprise to actually realize that an embryo at this point of gastrulation is quite different between a mouse and other mammals. And so we use the pig as a mimic or as a proxy for human development, because this particular period of time is very difficult to access in non-human primates, or never mind in human. We believe that using the pig as a model offers a great entry point into understanding what might be happening in other flat disk embryos. And so the pig, in a very accessible embryo, just to gather embryos from, makes it very suitable for these type of questions. And also the good thing is that pigs also implant quite late. So gastrulation is something that you can separate from placentation. That's not a trivial point. There is a lot of issues regarding the impact of the extraembryonic tissues in controlling how the fetus itself develops. So we are now working with an organism that undergoes gastrulation before implantation, very different to humans and mice. Really interesting side note that does have relevance for certain lineages. And I'm not going to talk about that today, but I think there is a lot of things to consider within that. As you can see also, obviously, the mouse is conical structure, very different topology, very different cell interactions when these major decisions are being made in an embryo. So you can imagine that some things might have adapted rodents that are quite peculiar or proper to them, might not help us really understand what happens in a conventional flat disk embryo. Having an equivalent can be informative, we believe so. And so I'll show you an example today of something that we show that it is that we think applies to humans. So the period of time that we are dealing in our work concerns, you see in the dark green area here, that's the period of gastrulation in a pig. It's around 11 days to about 15 days. So in a human, this would be anything between day 14 to about day 30. But the only data set available today from a human embryo is from this stage, very kind of advanced stage of gastrulation. We don't have actually a clue what's going on at this point. And in monkeys, very limited information exists around this area. We can divide gastrulation into two broad processes, primary and secondary gastrulation. Primary refers to the the formation of the three major germ layer progenitor cells, which is depicted here in light green. The darker green reflects the secondary gastrulation, which is the axial extension, where the embryo starts. Okay. These two processes can be divided and studied in great detail in the mouse. As you can see, the entire section of the mouse has been profiled death. There is nothing escaped. For other species, it's quite limited. So we try to fill the gaps using the pig embryo as a model. And so far, I think we have, now with the rabbit, I need to add the rabbit here now because the rabbit was published last year, has also quite a comprehensive coverage of this. So as I said, the detailed data sets are limited in other species. And for human embryos, we only have a couple of thousand cells that we can really work with. So really limited information. So we think that using these systems, these comparative embryology approaches will help us understand questions relating to heterochrony, the differences in the pace at which things take place in an embryo, then address questions about speciation. What are the peculiarities of different mammalian species? We think that there is a lot of scope using these single-cell technologies to understand it. So last year, we published the first atlas, comprehensive atlas of gastrulation in the pig. And this really nicely offers us a template for doing all the work that we want to do in vitro, but really in initial understanding of complexities and how the different lineages are emerging during this period of time. So you can see here in this plot, 36 cell lineages that appear at different times. You can then start looking at the interactions, the relationships between the different cell types. Does the blood emerge? When do the muscle progenitors emerge, and the different types of mesoderm and endoderm, etc. This is a nice, we're going to be using this quite a lot in the coming years, we believe, and there will be similar ones equivalent appearing in other species, we hope very soon. From this, we can already start doing some comparisons with other species available data sets. Particularly the mouse, we've done it. Obviously, we match everything to the mouse with great detail because the detailed data set available. We've also done some alignment, cross-species alignment with the monkey data sets. These plots here show you the correlation scores between the dates of the sampling of the different species. And you can then start mapping out when does a mouse 8.5, what does it correlate to with the pig? And so that's around E14, highest level of correlation. And the similar things can be done with the monkey. So now we can start having unmarked dates where we can start in meaningful comparisons and studying cell lineage allocation in these different species. So because from the human we are limited, we need to come up with a way to study this, hopefully in alternative species and also using in vitro systems. So this is the problem that we have. So that human embryo cannot really be accessed in a meaningful way for any functional work or even the work. Then we propose to use some similar species for then the pig embryo offers a very good entry point. We have then the potential to using stem cells to model some of the aspects of development in the pig embryo, called the gastroid system. And then we can model the equivalent gastroid system using embryonic stem cells in humans. And then when we have these two together, we can always begin to infer that some of these events might be taking place in an embryo based on the similarities between the in vitro systems and how they relate to the pig embryo. This is roughly how we try to operate and extract the most information possible in this system. So the good thing is that we can integrate, therefore, the use of vivo material with the application, the usage of embryonic stem cells, which come from these equivalent stages in vivo. And then we can begin to model and generate these 3D systems where we can start modeling the anterior-posterior patterning that takes place during gastrulation and looking at the complexity of the different tissues that give rise when these structures are made, and ask, do these things resemble anything that happens in an embryo in vivo? I'll give you an example of a scientific question that was pending and quite topical a couple of years ago when we were doing this work in terms of how does a bilaminar disc embryo allocate cells towards the endoderm lineage. And this is the textbook diagram of how the endoderm emerges in the mouse. And what you have is an epiblast which undergoes the epithelial to mesenchymal transition to create It's in the primitive streak, which eventually splits into two groups of cells. Cells on the top will give rise to the definitive endoderm or the gut progenitors, and the rest makes the mesoderm. So that was the question. And there was recently an alternative model proposed based on observations actually in the mouse. So it's been very contested in the mouse, whereby epiblast cells, rather than dividing into two groups of cells, they actually make what we call the anterior streak cells, which give rise to the definitive endoderm. Then another branch gives rise to the more posterior side of cells making the mesoderm. And then an area that gives rise to the notochord slightly later in development. And this comes from this study, a study published in 21 by a group in Germany, Michael Lickert's lab, who's observed that cells in the anterior part, this is the structure of the embryo, that's the epiblast. The purple cells here are the mesoderm. And he described an epithelial to epithelial transition in a very small number of cells, about 5 cells in a mouse, can give rise to the endoderm. And he called that an epithelial plasticity. There is evidence that some of the contribution of the endoderm comes also from these cells here, what we call the mesendoderm, these progenitor populations. That's what has been the long-standing paradigm in the mouse. We have done sections of pig embryos. This is a top view. This is a cross view of what the anterior-posterior sagittal section looks like. And this is in a real, actually, section of an embryo. At the particular stage when the primitive streak is forming, you have a group of cells here, the top side of the epiblast, are labeled for SOC17, which is a marker of definitive endoderm. Cells appear here in the anterior part. We observed these cells a long time ago when we published this paper, which was linked to something else, but this was side observation. So we should be exploring this. Because based on Michael Lickert's lab, suggests that maybe these cells might be representing the cells that he's describing here. Just never noticed and never put any effort to understanding this. Maybe this event is described as a rare event in a mouse, really as a secondary event. Might actually be taking place in an embryo that looks more like that this can. We set to test this using our single-cell transcriptome. So now we have a time course that we can access and interrogate day by day. So the first thing that we did from this subset of 100,000 cells that we had, we focused on the mesoderm and the endoderm lineage here in particular. Markers that indicate whether the cells are undergoing EMT, epithelial to mesenchymal transition. It's an indicator of mesoderm formation. What we find is that the cells in this branch, which make the mesoderm, have upregulation of SNAIL, which is a typical marker of EMT. The cells that branch to the right-hand side, which make actually the definitive gut, they actually maintain EpCAM and cadherins, which are both markers of epithelial cells. So that means if you are in epithelial cells, you normally don't express SNAIL. And at the same time, we saw expression of FOXA2, which is a typical marker of definitive endoderm, as well as SOC17. So these cells are branching very early from the epiblast. And actually, if we break this down by day, we can see that the cells that are labeled here in bright green, turning towards the left here, are the cells that are expressing FOXA2, NANOG, SOC17. For those who are not familiar with these markers, NANOG is a marker of an epiblast cell. So the stem cells, the cells in the epiblast express NANOG. FOXA2 is exclusively expressed either in the hypoblast, which is a current tissue, or the definitive endoderm, as well as this SOC17. What we see is a branching taking place early on at 11.5 and then ceasing at around 12.5. So it's a very temporal definition of this position in terms of giving rise to the definitive endoderm. You wouldn't see that if you just bulk embryos and you just look at the entire thing. You need to look actually day by day. And then later, where you have the emergence of notochord, node, is the third branch in this alternative sequence of events. So it's all happening a sequence of time, early and late. And when you look carefully in an embryo, so this is a non-spatially defined result, right? It's just a group of cells being aligned with an algorithm. But you go back to the embryo and you stain for these markers, you can see that this is the anterior part. That's the posterior part of the disc. The primitive streak will be forming here. This group of cells here, the green cells, are the ones that are labeled with FOXA2. They are just in front of the cells in the mesoderm. They only appear at this particular time. Later, they're gone. Actually, if you do a cross-section of this embryo, you look at this with the confocal microscope sideways. This is the anterior, that's the posterior. This is a close-up of this. You can see the cells. This is the epiblast. This is the hypoblast. Intercalating cells are NANOG positive cells. NANOG is not expressed in the hypoblast. The only reason why you have a NANOG cell there, we believe, because these cells have come from the top, just delaminating and going down. Just didn't catch one. We will have movies of this in the paper, but we can look into detail. But the point is that we present this as evidence that these NANOG cells actually are FOXA2 positive cells that are coming cells on the top. Now you look at that a day later, half a day later, these cells are gone. These cells are no longer expressing NANOG by the time you push this by another day or a day. It's a very temporal resolution of this and dependence. And what we wanted to figure here is, why does it happen in that particular window of time? No earlier or no later. There's a reason for that. This is the model that we had to explain how we believe the endoderm appears in the disc embryo. So we have the emerging mesoderm at the back, which produces Wnt. Wnt is an important source of signal that tells cells to do things. In this case, it's EMT. And then nodal is another signaling factor coming from the hypoblast. It produces in great abundances. So when you have a sweet spot of combination of Wnt and nodal in the right place, you induce expression of FOXA2. And these cells are then delaminating. And basically, you have different shading of green here. My postdoc is very artistic, and he likes playing with nuances. So he suggests that these cells are all becoming now definitive endoderm. When this happens, the nodal signal goes down. That's why you stop making endoderm at a particular time, because you're diluting the effect of nodal. So can we prove any of this using an in vitro system? Well, we turn to gastruloids, which is these models of gastrulation that can mimic some of the events. And so essentially, these are aggregates of cells capable of symmetry breaking and lineage specification and axial patterning. And they can basically be used to study the body axis, the anterior-posterior elongation, and they actually collinear expression of the Hox pattern. That means that they have an anterior-posterior identity, and they were described first in the mouse, more recently in 2020 by the Morris lab, Alfonso's Pinezarias' group in human. And the beauty of this is that they are very high throughput, amenable for manipulation, and you can do then comparisons across species. Because this is the ones that we're making in pig now. They look really very similar to the cow, the human. We wanted to use this system to explore that question. And we actually figure how these cells, presumably in the posterior part, induce the cells in the anterior part, become specified into FOXA2 cells. So we've mixed two lines of cells. One which was exposed to Wnt. These cells equivalent to this. These stem cells are exposed to Wnt overnight, then mixed, as you can see, these two groups of cells in the same tube with GFP-labeled embryonic stem cells, which basically represent the epiblast. The epiblast is a stem cell in the presence of low levels of activin. We make these aggregates because it's a very early event. We didn't wait 3 days. We just waited overnight. So we just wanted to see the effect after a 24-hour combination. So that means that the stem cells, which should be labeled as GFP cells, are labeled with green in the cytoplasm. The GFP protein is just ubiquitous. And we see a cluster of cells emerging after 24 hours expressing SOC17, which is a marker of definitive endoderm. That means that the cells that were neighboring the purple cells are the cells with chiron, the danalo, were induced to push directly overnight, this is 24 hours, into SOC17. We believe that this is recapitulating what I showed you in the embryo as cells that are switching on FOXA2 and SOC17 in a very time-resolved manner. And this is in response to the Wnt activity, which is produced by the cells themselves. The cells produce Wnt in this particular location, and they are inducing the neighboring cells to activate this. So this is a good example of how gastruloids, the early gastruloids, can begin to address some of these questions. Since then, we've been doing additional work on gastruloids to study additional questions that obviously emerge once you have cell lines from different species. In the lab, we have pig, cow, sheep, human and mouse. And so we want to use this as a way to address multiple questions. This is a good review that tells you how you can use them. In our lab, we're particularly interested in the idea of speciation, how special features emerge over time. And this idea of the hourglass was proposed many, many years ago by Denis Dubourg, who wrote a review about his idea from 1994, where he describes a period of development where there are developmental constraints. So every animal passes through that without really messing around too much with its genetic makeup. And that's not really been formally tested, really, in mammals at least, in great detail. We don't know what the phylotypic stage is at genetic level. We want to start using gastruloids to start addressing some of these questions. We have a system where stem cells can be exposed to a source of wind, which posteriorizes the cells. They resemble the posterior part of the embryo. There's a little movie that shows you how these structures actually look like when you look under the microscope, and they are stained for mercury, which is a marker of the posterior side of the embryo, and SOX2, which is a marker of notochord in this particular stage of development. See a neural tube forming here in the middle of the embryo. These structures are really very anterior-posterior patterned, and you can begin to interrogate what else is in there that resembles an embryo. So the top figure here shows you the entire map of the real pig embryo, and the cells that you see here labeled in the bottom right are the cells that were identified by single-cell transcriptomics in this object. And you can see that there is a good complexity, good mixture of cells. There is a lot of cells from the mesodermal layer. Makes sense because these cells are induced with chiron, which means it's a posterior section. It doesn't make head. This gastruloid doesn't make a head because it's induced to make posterior part, the tail of the embryo. And notochord lineage. You can see here this branch here, which makes the notochord. So all of this is represented within the gastruloid. So now we can start asking questions about the posterior side of an embryo in general terms. When we do RNA-seq of these structures on a time course of these 24, 48 to 96 hours, you can see clearly how they change. They become anterior-posterior patterned. There's a lot of apoptosis taking place, which actually happens in vivo as well. And we start looking at the general expression pattern of this in a bulk fashion, and we can see expression of these groups of genes. You have even Hox expression, neurospecification, notochord specification. So these are really beginning to develop. And you can do then cross comparisons with data sets from human and mouse. And in a PCA plot, you can see here that there's a time course relationship between time and species. Most of the time, in most cases, there is a really interesting relationship between the species and what happens during this gastruloid system. So there is also a scope for species comparison. But this is bulk to go single cell to really gain much detailed understanding. So we have done this experiment across 3 animals. And these are how the gastruloids look like after this time course. These cells, by the way, are all grown in exactly the same conditions. This is an important detail. We can go into the questions later, but some of the comparisons have been published are widely different stem cells. So it's very different to come up with a meaningful conclusion to really what's the heterochrony here. Here we're comparing cells grown in exactly the same conditions and exposed to the same treatment. That means if they look different, there must be something inherent in those cells that might help us explain the difference. One of the things I want to point out in this figure, which, broadly speaking, they do look similar. The cow is a bit weird. It has some more extreme phenotypes. Look at the scale bar here. This object is very large, fit in an image, but basically much bigger than cow and human, and much more contorted. That's very, very strange how gastruloids actually behave. But we have done single-cell RNA-seq. We have superimposed the gastruloid map, the pig. There is no bovine atlas. The closest we have is a pig atlas for studying the cow gastrula. And broadly speaking, everything is there. There are some differences, and you will see here the spinal cord. When we look, for example, at the Hox profile between the 2 species, broadly speaking, they have the Hox clusters acquired. In the pig, they appear a little bit earlier in development. It seems like the pig is in a rush compared to the cow. We also broadly recapitulate what we see in vivo. So if we map our single-cell data from the stem cell to the 72-hour period, we map quite well the embryo data. quite good shift. Perhaps the cow is a little slower. It just transitions a bit slowly between 14 and 15. The pig is more presenting 15 days at the end of the process than a cow. It's not surprising, I suppose. And also, well, there's an issue with the title here, but never mind. The formation of specific lineages. So these are the number of lineages we have. It's about 12 or 15 lineages. What we see is the cow and the pig have somatic mesoderm, thermomyotome, and sclerotome after 48 hours, whereas the cow, a lot less of that. And we also have very limited evidence of spinal cord formation in cow at 48 hours. So the spinal cord is this blue area here. The spinal cord is quite a late tissue. So this gastroliths actually can go quite late in the pig. But in the cow, this seems to be a little bit late. It takes an extra day to get there. I think we are getting to some sort of approximation that there is a potential model here to study heterochrony. Why do these cells be longer? Is it proteolysis? Is it ubiquitination? Is it protein? We don't know, but now we have a system we can start interrogating. And we thought, well, let's try playing with the signaling within systems. Can we tweak things and make these shapes change? So that's how a conventional pig gastrolith looks between 48 and 72 hours. As you can see, it's a very oblong structure. It has an anterior-posterior pattern, and it does a constriction. When we add a nodal inhibitor, just for a day, between 48 and 72, you can see quite a shift in its structure. And we don't know what dictates that. What have we messed with in terms of cell type that has led to that? But it's important to remember that we are comparing species. This doesn't look too different to cow gastrolith. So the question is, can we start playing with signals and one species into another? This is very interesting for us in trying to understand heterochrony for precisely these reasons. Once we understand the signals that dictate this, which might be entirely driven in vivo by extraembryonic tissues, this is totally pure, devoid of any of that. So we can really interrogate specifically signals that might come from different parts of the embryo. Might explain why a cow looks like a cow and a pig like a pig. And so, and why? Why is that important? Well, it's important for many reasons. First, to understand the way that programs have evolved over time in different species. So we have different ways to interpret how cells appear and disappear, or how progenitors emerge and how subsequent cell types appear in a sequence. There are multiple possibilities. It could be that the differences between species are down to more rapid development of exactly the same profile of cells. So if these cells give rise to these two cells and these branch out and give rise to sequence cells, that takes a certain amount of time. It might be that in a different species, let's say this is a pig, this is a mouse, exactly the same branching takes place in a much shorter time. That's one way of interpreting. Alternatively, no. Alternatively, it's a situation where the branching takes place in a different order. So not only is it longer in a pig than it is in the mouse, but perhaps the branching itself changed. We don't know any of that in mammals. We are keen to interrogate this using this cross-species comparison of gastrulation as a window. It does suggest that there are some differences of that nature. We have seen differences in, for example, the emergence of the hematopoietic lineage between cows and pigs. But we need to go back and validate some of this in vivo, because in the cow we haven't got data set. I think these are interesting hypotheses that we can start interrogating the in vitro system. And the reason I think it's important, from a more practical side of view, is that developmental tempo is important to understand how organs develop. That's important in experiments such as this one. It was reported by a group in America who introduced human cells into a pig embryo. This pig embryo was edited for the 6-1 gene. Basically, these animals didn't have a kidney unless human cells rescued the kidney of this pig. They did. So chimeric embryos were made of human kidney. The remarkable thing about this paper is that at day 35, this is the mesonephros of this SAL1 no rescued human ES cell embryos. Normal kidney. A normal kidney in a human doesn't form until a week or two from this date. That means that human cells were informed by its environment to accelerate the developmental program, give rise to, presumably, according to the description of this paper, a very normal kidney. But that means that you can manipulate in the right environment. You need to understand how that works, what the network is, and how to control it, basically. Because the application, as you can imagine, would be quite enormous if you could actually do this and produce normal organs. But you need to understand the underpinning basis and the biological basis of organogenesis. We think there is a lot of interesting questions that can be addressed with this system. And basically, it's late. I'm not going to finish this by saying that we believe that cross-species comparison of atlases across different species can be quite informative to inform cellular trajectories. Gastroloids are incredibly valuable models for investigating this critical stage of development, understand some principles that apply across species. That this will help us develop more interesting strategies for example, these chimeras and hopefully the generation of organs for disease. Just to acknowledge the group members, so Luc has been primarily involved in the Atlas work with Doris, with my experienced lab technician who does all the stem cell work, and the rest of the team for helping us, and the collaboration with Alfonso, it's been quite instrumental in developing the cross-species gastroloids, and our funders for their support. So thanks very much. Happy to take questions.
Questions? I know we are a little bit late. May they have any questions? Or
I have two questions. The first one is linked to the inductions of SOC17 cells by the chiron-treated versus stem cells. Have you seen the loss of nanog in this situation? And it's come back to your previous pictures when you see the intercalation of apoblast cells and definitive endoderm cells from the epiblast, where you have still nanog. What is the time where the cells are going to be similar? You mix the two populations. You are going to lose nanog expressions and to get a final endoderm fate.
So in the asteroid experiment, we stained for SOC17 at 24 hours. By the time that happens, there is no nanog. So you never see nanog and SOC17 co-expressed. So it's a quite definitive phenotype, if you like, a quite terminal phenotype, SOC17. Deliberately chose SOC17 because it's a more unambiguous marker of definitive endoderm. And when those are double stained, they have no nanog. And that happens in 24 hours. It's fairly quick. Switch. Vivo, that might take maybe a little later. This is kind of forced system.
My second question is linked to the gastroloid part. When you inhibit nodal or TGF-beta, you're inhibiting laterality. Can this explain the fact that you have a twist and that it may regulate forces within the embryo?
I think so. We don't know yet. We haven't sequenced it. We have collected the sample. We just the phenotype, and we think there is an issue with the mesoderm lineage, laterality that might explain this. But it's just, yeah, I was curious to see such a drastic effect in such a short time. We will explore all these questions when data comes.